Thank you all for being here and traveling to the great town of Manhattan, Kansas. I am honored to be here today to present, to be invited to present at this conference. I'm humbled to be speaking between John Blair and Kelly Kincher, but most of all, I'm excited to share with you some of the long-term research that I've been doing at the Prairie Biological Station and the insights that I think it has for conservation and achieving conservation goals. Those goals of increasing biodiversity, promoting resilience of these ecosystems to periodic stress like drought, and all of us trying to anticipate ecosystem and community responses to climate change, and the potential to enhance landscape heterogeneity and biodiversity, all of which require human intervention. I just saw the screen down here. So as most of you know, many of you know, conservation and ecological restoration are human interventions. They are human efforts that aim to influence ecosystem state changes, to prevent ecosystem state changes and transitions that John was talking about, particularly here in grasslands. So at the landscape scale, that human effort is often effort working towards prevention of undesirable state changes. And these efforts are operating on the social system. Land that is designated for urban development, land that is going to be used or is used and how it is used for agriculture versus land that is managed for grassland bird conservation. At what I'd like to refer to as land ownership scales, human intervention is management often being used that all of you in here are probably using to prevent undesirable state changes. And then at various scales, we are often reacting to these state changes and exerting effort to reverse those state changes to something that is more desirable by ceasing drivers that cause those state changes. And ultimately, science and stewardship should work, and hopefully will increasingly work together to inform not only our understanding of nature, but how we can achieve these conservation goals. So again, John Blair talked about the various state changes that have happened even within the property of the Prairie Biological Station by experimenting with varying ecological drivers on the landscape to result in maintaining grasslands, development of shrublands, and development of woodlands. But he also mentioned another major state change in grasslands globally, as well as the Tallgrass prairie ecosystem, and that is its conversion to agricultural use, which also requires human intervention to reverse, to reinstate the species that used to be there, and this is even more critically important in areas where prairie is highly, highly fragmented, ultimately to achieve a state of a desirable grassland state. And so that's really the system that I work in at the Prairie Biological Station. So today I'm going to talk about two studies that have been going on for decades now in the lowland agricultural lands at the Prairie Biological Station. One is a quarter-century application of an ecological theory to see if we can increase biodiversity and ecosystem functioning and resilience of prairies that we restored. The second is a decadal setup and multi-decadal study of restored prairie response to interannual variability in planting year climate in hopes to gain some information about anticipating community change in our increasingly variable world. So the tallgrass prairie ecosystem is inherently heterogeneous, and John Blair laid a really nice foundation for your understanding of causes of that heterogeneity. So we know that the heterogeneity in communities and on the landscape is maintained by variation in topography, as well as the frequency and intensity of ecological drivers like fire and grazing. And there are a number of studies at Kanza and at other grasslands that show positive relationships between plant diversity and heterogeneity in soil resources. And so most of the tallgrass prairie, again, has been converted to agriculture, and traditional agricultural practices like conventional tillage, we know, have homogenized the soil environment for decades, and this is a starting point for many prairie restoration efforts. So we had a pretty simple hypothesis, and that was that enhancing heterogeneity in the soil environment would promote plant diversity in restored prairie. The theoretical rationale or basis for this hypothesis is grounded in the environmental heterogeneity hypothesis that predicts spatial variability in resources will increase the availability and variety of niches for more species to use and enable their coexistence, and as a result to increase biodiversity. So we started this experiment, or setting up this experiment, in 1997, where we performed some soil manipulations, fairly minor soil manipulations, in an agricultural field. And so that at Kanza Prairie, that had been cultivated for 50 years. And so this site at Kanza contains 16 different experimental plots that vary in the amount of soil heterogeneity. So we had 4 treatments. We had control plots where we didn't manipulate any soil resources. We had plots where we had alternating strips of buried limestone to create those deep and shallow soils, because we know that soil depth and topography are an important driver of diversity and productivity in tallgrass prairie. Plots where we altered nutrient availability by having strips where nutrients were just left at ambient conditions, strips where we increased nutrient availability by adding fertilizer, and strips where we reduced nitrogen availability by adding a labile form of carbon to pretty much to increase the microbial biomass, and those microbes suck up the available nitrogen. And then last, we had these maximum heterogeneity plots, as if you overlaid the nutrient heterogeneity and depth heterogeneity plots, and I'm going to focus mostly on those today. And so after we imparted all of these treatments and we maintained these treatments over time, we initially seeded the site with 42 different native prairie species. And so the first 3 years of this project, I'm going to give you a short version of a pretty long saga. There was no effect of these heterogeneity treatments. So any of you who have restored prairie on the landscape have experienced a huge flush of agricultural weeds that happens in that first year. And then going into this particular restoration, the third year, the field was pretty much a solid field of switchgrass. But there were, underneath these layers of agricultural weeds and dominance of switchgrass, there were some pretty significant diversity responses to resource availability. So we found that diversity was inversely related to the amount of nitrogen that was available for plant use. It was also inversely related to above ground net primary productivity, and positively related to the amount of light hitting the soil surface. And in fact, you could see some of our treatments, the carbon addition treatment here in this picture was just surrounded by the agricultural weeds that prefer more nitrogen. And we found really strong diversity responses to these nutrient treatments, where we had highest diversity initially, where we reduced nitrogen availability in the soil, and lowest diversity in our enriched nitrogen treatment. And if this experiment had been conducted somewhere other than a biological station or in a long-term ecological research site, that might have been the end of the story. So in 2005, which was the 8th year of this experiment being in place, I had moved to Southern Illinois University. I was interacting with practitioners at Natusa Grasslands, had a graduate student who worked at Natusa Grasslands, got the idea of overseeding and adding more propagules to increase diversity. So that's what we did. We added 20 new species to the entire experiment. And it followed plant diversity and community responses in the heterogeneous and control plots for the longest period of time. And so looking at the 15-year community dynamics in this experiment, we found that species diversity, the log relationship of diversity with time, that diversity declined over time, plot level species richness declined over time, but we saw a lower rate of decline occurring in the heterogeneous soils. And the reason being is when we performed that propagule addition, that bumped diversity up in the heterogeneous soil treatment compared to the control that seemed to go away about 6 to 7 years later. So that inspired us to pursue more funding and do more propagule addition, repeated for 5 years, and then to follow these communities for 5 years. And so we hypothesized that with this continuous propagule addition, that would offset the declining diversity in this system, and it would be to a greater extent offset in our heterogeneous soil treatment. And that heterogeneity would promote plant diversity, and that higher diversity will promote ecosystem functioning and resilience that is predicted by biodiversity and ecosystem functioning theory and diversity resilience relationships that have been seen in other grasslands. So when we look at just the period of propagule addition, so now we're looking at the 20-year diversity dynamics, we see that when we added these propagules for 5 years, we indeed did increase diversity and richness in the soils that had these heterogeneity treatments. But what about afterwards? Afterwards, after this 5 years of propagule addition, we see that the heterogeneous soils are actually maintaining higher diversity and higher richness. And when we model the dynamics of diversity over this quarter-century period, we find an exponential decline in diversity in our control soils that this model no longer fits in our heterogeneous treatments. And when we look at the time of adding propagules and 5 years later, we find a continuous decline in species richness in the control soils that the heterogeneity offset in the presence of propagule addition. We also compared productivity in these treatments to native prairie. And the productivity was collected using the same methods in our experiment as it is in other lowland prairie experiments at Kansa Prairie to analyze whether or not we have restored functioning that is similar to native prairie, and also examine whether or not our restored prairies are as resilient to drought. So we were fortunate to have a collection of above ground net primary productivity data that spanned a pre-drought period, the presence of a pretty severe local drought in 2018, and measurements after the drought. And so what we found was that when you look at the average productivity in the non-drought period, the productivity between our restored prairies and native prairies is not any different. And in fact, the productivity during the drought of 2018 is also not different between the restored prairie and our native prairie. But after drought, there's actually a greater productivity response in the restored prairie. So both prairies types have a positive drought legacy effect, higher productivity after the drought, but it's higher in our restored prairies. And so when we break down actually the different native prairie sites that we sampled and parsed our restored prairie productivity samples into the homogeneous treatment and heterogeneous treatment, we find that the higher productivity post-drought response in our restored prairie was actually due to the response in the maximum heterogeneity treatments. So some of the lessons or insights for conservation from this study, I believe, is that we do have some solid evidence that heterogeneity can promote biodiversity, ecosystem functioning, and measured as resilience to drought, and that the effects of heterogeneity on plant diversity can take decades to actually manifest, and particularly in communities that are dominated by clonal species, and for these effects to emerge may require the addition of propagules of new species, so continuing to work with those lands that we are trying to restore for biodiversity and functioning. So now I'm going to switch to another long-term, or becoming a new long-term study in restoration at Kansa Prairie. And this study is motivated by, I think, many practitioner concerns, or concerns in the conservation world, is how do we prepare for understanding how the communities that we are managing and restoring, how are they going to respond to climate change? And I don't have all of those answers, but we are at least focusing on how to anticipate some response to variable climate. The rationale for this work is based on the fact that we do have fairly limited prediction in restoration ecology, and in its attempt to move our science towards prediction. It's also based on understanding that variation in the environment can result in the assembly of different communities. This is a phenomenon referred to as year effects. And so, with knowledge that these year effects occur, if interannual variability in climate is a deterministic driver of community development, then planting year climate, we might be able to use that to predict restoration community outcomes. And if we have knowledge of whether there are going to be wet years and dry years, how can we prepare to restore resilient communities with that knowledge? So we refer to this study at Kansa Prairie as the sequential restoration plots. So this is an agricultural field that we have taken 4 plots or strips out of production and restored to prairie every other year starting in 2010 and ending in 2022. So it's taken 12 years to set up this project. We've attempted to control as many factors as possible. Those factors being controlling this, using the same seed source, collecting most of the species from Riley County, using the same species in all of our restoration mixes, getting every species every time we perform a restoration, tested for live seed, so we use the same live seed number for each species. And again, we're in the same field that has the same crop history, that has the same soil type, and we use the same restoration methods. So what we're really doing is gambling on the weather being variable in Manhattan, Kansas. And that's a pretty good bet. So in 2010, actually quite surprised in 2010, because the weather is very variable. Kansas' average mean precipitation is like 835 millimeters per year. That is hardly ever observed, but we observed that in the first year of the sequences. That was followed by putting a restoration in during the extreme drought of 2012. And so we see variation in total precipitation, but we see really a lot of variation when we're looking at precipitation and climate in June and July, which is a critical period for seedling emergence, especially when we were seeding these areas at the end of May each year. And so just to give you an idea of what the sampling regime looks like, of course, we establish these prairies every other year, but we also follow them, we survey them through time, right? So the oldest prairies, we have the most information relative to the newest restored prairies. So the first study that came out, we looked at prairies that were established, 3 different prairies established over a 6-year period. Each of those followed for 3 different years, for their first 3 years of community development. So this is a multi-metric dimensional scaling graph, basically showing each dot represents whole plant community composition. The closer the symbols are in this 2-dimensional space means that the closer those communities are in composition. The different colors in this graph represent the different sequences or prairies when they were restored, and the lines and symbols represent the trajectory of change over time. And so what surprised us when we just initiated these kinds of analyses is that all 3 prairies in the beginning, restored in 2010, 2012, 2014, started it with significantly different composition of species. They changed through time, and in the 3rd year, they still had significantly different composition of species. But the ones that were restored in 2012 and 2014, slightly wetter years, were a little closer together in this ordination space than the prairie that was restored in drought. So moving on in time, and students as well, there's a lot of generations of students who are accumulating during this project. We analyzed the 2 sequences for which we had the most long-term decadal data set. And so what we found when we did that, comparing the kind of average year with the drought-restored prairie, is not only did they start compositionally different, they remained compositionally different for a decade. And that really surprised us. And you can see in this ordination space that the communities restored in these different years are sort of stabilized in these different, significantly different composition of species and their relative abundance. So the burning question is, what is different between prairies restored in a wetter average years compared to drought years? And so surprisingly, we find that the prairie restored in drought year, most years for the next 10 years or 9 years, has higher species richness and higher cover of prairie forbs. So the most recent study, this is fairly hot off the press, master's student who just finished, was to look at, now we have many prairies going in up until 2021 and 2022 that have been restored for 3 years. So now we have enough information over enough variation in climate to attempt to model whether communities are predictable by planting year climate. And so our hypothesis here was fairly simple, not so simple to analyze, was that we hypothesized that the greater dissimilarity that there was in planting year climate would result in greater dissimilarity in communities that were restored. And so up until 2021, we only had 6 sequences that were restored for 3 years, and so that's the data set that we used. And so we compared community composition among all of the subplots to each other across sequences and within a sequence. And we were very excited and surprised to find that we found this relationship. We looked at several different climate variables, but the strongest predictors of community dissimilarity were climate variables in June and July. And so when we look at this graph, you'll see a cluster of comparisons on the far left-hand side where dissimilarity in climate is 0. That is communities within a sequence being compared to themselves, right? As opposed to other sequences restored in other years where you have greater dissimilarity in climate. And even when you take away the self-comparisons, that relationship remains significant. So it's not driven by just the 0 on the x-axis. And so we see a positive relationship with June and July precipitation and June and July temperature. We see these in the initial year, not in the second year, and the relationship is stronger in the third year. And so the third year is significant here because this is when most of the annual agricultural weeds, volunteer species, have fallen out of these restorations. And to corroborate what we found in the study looking at just comparing the wet restored and the drought restored prairies and the higher forb cover and higher richness in the drought restored prairie, now we can model grass and forb and species responses to climate across this planting year climate gradient. And our results from this corroborate what we found from just that single comparison, in that the grasses and their relative cover is strongly and positively related to planting year precipitation and inversely related to planting year temperature. And we find the opposite responses with the forb cover. So if any of you read Chris Helzer's blog, he recently had a post about heterogeneity on the landscape, and that's what one major conservation goal we try to achieve. And many of you might be working on fairly small landscapes. You might be working in small, relatively small fields and with some projects. But this study nicely shows that doing these temporal sequence of restorations increases heterogeneity even at small scales on the landscape, as opposed to restoring a site at one point in time. And of course, we see this at Nachusa grasslands as well. But when we look at the data of species richness, combining all of the different temporal sequences of restorations, if we look at all of the sequences in their first year, all of the sequences in their second year, all of the sequences in their third year, we don't see the decline in species richness. we actually see that the collectively richness increases with growing season, number of growing seasons from 67 to 95 to 102 species. So we can have a positive effect on biodiversity by doing these sort of temporal installations of restorations. So what are the insights from conservation from this study? One is that I think we are learning that planting year climate has predictable effects on fairly coarse measures of plant composition, like the percent cover of grasses and forbs, and also on richness. But we know that whole community composition and that dissimilarity in communities is also strongly predicted by planting year climate. Second, we are finding that interannual variation in climate has decadal consequences for community composition, and again, being able to anticipate wet or dry years. With that knowledge, maybe we can modify seed mixes to achieve whatever that restoration goal is. And that variation in these plantings can result in greater biodiversity and heterogeneity on the landscape, which we have pretty strong evidence building that heterogeneity is important for maintaining biodiversity and ecosystem functioning. So with that, again, this has been a collective effort. I've collaborated with John Blair. I've had many students contribute to these projects along the way. It wouldn't be possible, I think, without being at a biological station or LTER site. And much of this research has been funded by the National Science Foundation. And with that, I think I have a minute for questions. Yeah, in the back. Okay.
So I really appreciate the length of these studies, and I think we've got to, I know there's challenges, but we've got to get there if we want to start getting some answers. You might have mentioned it, but in both these studies, what was the management of these plots after these seedings?
Yeah, that's a really good question, and no, I didn't manage it. So the first study, the site has been annually burned most years. A few years we've let it rest, the heterogeneity study, and that annual burning does promote grass dominance, but of course we know it's pretty much needed to prevent trees, and we're in a lowland area where there are a lot of sources. The second study is kind of interesting. We used much higher forb seed in our restoration mix, and in the first 3 years, actually, there was not enough fuel to burn. So the first 3 years had no burning, but then after that, they are all managed the same, and then they are annually burned to prevent that woody encroachment.
Okay, great. Yeah, I would think, that the addition of grazing, another ecological driver, would sure make a difference in maybe some of those findings of heterogeneity and biodiversity. And importantly, too, I think it might be yearly. Those plants will express themselves differently, maybe not long term, but I think that's all part of the discussion about biodiversity.
Yeah, I think we have a lot to learn in restorations and adding grazers. Unfortunately, they're doing that at large scales at Nachusa grasslands and collecting really important data on that. But a lot of our restorations are also happening at small scales and in small fields and conservation reserve program lands where it might not be possible to introduce grazers, but if any other kind of heterogeneity would be important. But yeah, there's a lot to learn about grazing and restoration.